Hi, and thank you for watching my poster video. My name is Nathan Bollig. I'm a veterinarian affiliated with the Computer Science Department at the University of Wisconsin. This poster is about adversarial mutation, a method for predicting the spillover risk of viruses from their genomic sequences. Spillover refers to the process of an animal virus moving into a human population, and efforts to improve the prediction of viral spillover are significant for pandemic prevention. Our work introduces a methodology that we call adversarial mutation, which uses adversarial machine learning to explore mutations that pose a high risk of spillover. We start with a model that has learned to discriminate between human and non-human infecting viruses, and a query sequence, for example, this bat virus. And then we use the model to search for mutations of that query virus that the model will rate as infecting humans with high confidence. The key here is that a sequence has a high spillover risk if this procedure can find a high confidence variant with a small number of functionally relevant mutations. We tested this method with a set of coronavirus spike protein sequences, some human infecting and some not. The first step was to learn a convolutional neural network-based model that can differentiate between those two classes. The area under the receiver operating curve for this model was 0.71. Adversarial mutation involves using that host classification model to carry out a search for high-risk mutational variants of query sequences. Given a query sequence X, the task is to find a sequence X', which is a mutation of X, that minimizes a cost function and has model confidence above a specified threshold. To do this, we currently use an iterative algorithm inspired by prior adversarial machine learning work in the text domain. We then used adversarial mutation to compute the spillover risk of SARS CoV-2 sequences, which were withheld from the underlying host classifier at training time. SARS CoV-2 sequences were ranked higher than they would be if we had used some alternative baseline methods like BLOSSOM similarity to the closest positive or the initial model prediction of the underlying host classifier. In the panel on the right, for example, the spillover risk of sequences is ranked by the initial model prediction on the horizontal axis and by our adversarial mutation method on the vertical axis. You can see that the SARS CoV-2 sequences, which are shown in red, are ranked in the mid-300s when the initial model prediction is used. However, when we use adversarial mutation, those sequences are pushed towards the top of the list, reflecting a high risk of spillover. We can also use adversarial mutation to characterize what types of mutations are most likely to cause spillover. In our experiments, the mutations most commonly associated with increased spillover risk occurred within the receptor binding domain of the spike protein. This region of the protein is known to be functionally relevant to the virus's ability to attach to and infect human cells. In conclusion, our adversarial mutation method repurposes adversarial machine learning for a spillover prediction task, and it allows us to rank the spillover risk for given viruses. In our framework, a high spillover risk is determined when adversarial mutation finds a high confidence variant using a small number of mutations. Our preliminary results have shown that ranking by adversarial mutation identifies known high-risk sequences better than certain baselines. Thank you for watching this poster video. I'd like to acknowledge the support of an NLM training grant and my mentors, Mark Craven and Tony Goldberg at the University of Wisconsin. Please view my poster or contact me for more information about this work. Thank you so much for listening.